My name is Amber Bartlett, and today I'll be talking about my project on soil respiration in a natural and restored wetland site in the New Jersey Meadowlands. Soil respiration is essentially the breathing of the soil, in which microorganisms and roots in the soil exhale carbon dioxide just like we do. Measuring soil respiration helps us to understand the carbon cycle, particularly the release of carbon dioxide back to the atmosphere, which is important today as atmospheric carbon reaches over 400 parts per million. Soil respiration in wetlands, however, is heavily understudied. It is now being acknowledged that wetlands play a critical role in the carbon cycle through the storage and emission of carbon dioxide. Despite only making up a small portion of our land surface, wetlands typically serve as a carbon sink, holding large amounts of carbon for long lengths of time, which alleviates atmospheric carbon from increasing further. However, repurposing wetlands for industry or other land use changes diminishes the chance of a wetland serving as a carbon sink and can even turn it into a carbon source. In urban environments, such as the New Jersey Meadowlands, where disturbances may affect their status as either a carbon source or sink, it is even more important to monitor carbon release through soil respiration. Our objectives in this study were to compare respiration in a natural and a mitigated site, and to also compare how different vegetation types respire. Our natural site had two species, Phragmites australis, which is a common reed, and Spartina patens, which is a salt meadow cordgrass. Our restored site had Spartina alterniflora, which is a salt marsh cordgrass. We randomly selected plots of vegetation in a natural and restored site using ArcGIS. We measured 11 plots of both Phragmites and patens in our natural site, which you can see here, and 9 plots of Spartina alterniflora in our restored site, which you can see here. We then placed a PVC tube into the soil and used an infrared gas analyzer, shown here. When placing the gas analyzer on top of the PVC tube, we create what's known as a closed chamber system, which just means that air is able to circulate in a loop around a CO2 sensor. We then allowed carbon dioxide to build up in the chamber for a cycle of 55 to 100 seconds, as shown here, and the flux was then calculated. The flux is a rate at which CO2 is being respired from the soils and microorganisms. Since we were measuring respiration in wetlands, where the entire area is covered densely in vegetation, we first measured respiration, keeping the surrounding vegetation inside the chamber. Then we removed the vegetation and kept it for biomass data, and measured respiration again with just bare soil. As you can see in this figure, when respiration was measured with just bare soil, Phragmites australis had the highest flux, and the lowest flux was with Spartina patens. Phragmites and Spartina patens are in the same site, so these differences cannot be attributed to the site, but possibly from plant and root structure differences, since one is a grass with tall height and the other is a short grass. When measured with vegetation inside the chamber, Spartina alterniflora and Phragmites australis had comparable fluxes, as you can see here, while Spartina patens was still much lower. The difference in flux with and without vegetation tells us how much the vegetation is respiring. The results show that Spartina alterniflora have the largest difference in flux from here to here, followed by Phragmites australis, followed by Spartina patens, and then Phragmites australis. However, when these flux measurements are normalized by vegetation mass, Spartina alterniflora and Spartina patens had similar respiration per unit mass, as you can see in this figure. When we measured Phragmites, because it is a tall grass, we removed everything above the chamber and only had the lower portion of the stem in the chamber. This might be the reason for the small difference in flux. Spartina alterniflora had a flux which was almost twice that of Spartina patens. However, when we normalized flux by vegetation mass, they had a similar amount of respiration per unit mass. And we didn't find any major differences in respiration between our natural and restored site. So, in conclusion, we found that soil under Phragmites is respiring the most. We also found that respiration of vegetation can contribute at least as much as the soil respires. In a future study, we would take more measurements so we can scale up our data to account for the entire wetlands ecosystem. This would give us the ability to compare carbon release in wetlands to other ecosystems. Thank you.